Hello, and welcome to Week 13 Basic Genetics. We will begin with a couple of reminders as usual. First of all, the case study assignment is going to be due this Saturday at 11:59. All of you have already been assigned an article to work with, so please take a look at the file and make sure that you are able to access your article with time. Week 13 PPP quiz is going to be due also this Saturday at 11:59. The quiz is already available. Finally, Fact or Fiction Group 7 is due also this Saturday at 11:59. So if you are in Group 7, you have all these three things due on Saturday. In addition to that, let me mention that there is also a new extra credit quiz, a week 3 extra credit quiz, okay? This quiz is going to have questions for Unit 3 exam, or the final now, pretty much. It is worth 3 extra credit points, alright? So you want to go in there and do the quiz when you can. However, understand that there is a smaller window of opportunity this time. We have been instructed to make this window 6 hours long. So the quiz is going to be due on Tuesday of next week, the same day that we usually have Lifeline. Depending on which class you have, if you're in the 6 p.m. class, your quiz is going to be open from 3 p.m. to 9 p.m. If you're in the 7 p.m. class, your quiz is going to be open from 4 p.m. to 10 p.m. That is 6 hours to do the quiz. All of you will get 30 minutes to complete the quiz as well. Now, these quizzes have been altered in a way that is going to match the professor's exam. For instance, you will not be able to go back a question whenever you answer a question. So please have this in mind as you are answering your questions. And keep in mind that it will be similar to the final when you're doing it, especially now that the final is more likely going to be on campus than in the Respondos browser. So let's get started. First of all, what is genetics? Genetics is the study of traits and their inheritance. The father of genetics is Gregor Mendel. He discovered the basic rules of genetic inheritance. Discoveries were made by breeding pea plants, studied the inheritance of traits, phenotypes, of an organism by observing the offspring. This is why the basic genetics are known as Mendelian genetics. Terminology. Some of these you have already heard, the others maybe not. First of all, by now it should be quite clear what a chromosome is. A chromosome is a condensed structure of coiled DNA present in all living cells that carries the genes determining heredity. So this is, if anything, a little bit different of a definition of what we have already seen. A gene is a specific sequence of nucleotides in DNA involved in synthesizing polypeptides. So, going back to the central dogma, right? DNA, a gene, pretty much, codes for RNA, which then translates into a protein. And that protein is what gives you your phenotype. Alleles, as we know again, they are alternate versions of a gene, which account for variations in inherited characteristics. For instance, you can have a dominant allele or a recessive allele. Now, this one is quite new to us. A locus, loci in plural. It is the location of a given gene along the length of a chromosome. So this is like the coordinate of where the gene can be found along the chromosome. So here you have it. These are different gene loci. And then you have a dominant allele here, and you have a recessive allele here. We will get more into that. Dominant alleles are variations of a gene that are always expressed and can mask a recessive allele. Only one dominant allele is required to express its corresponding physical traits. The dominant allele is present in an uppercase letter. So that is how we represent a dominant allele most of the time. If we use a capital letter next to a lowercase letter, we know that the capital letter is the dominant type. Now, both recessive and dominant have a phenotype associated with it. But this is the important part. The dominant allele can mask a recessive allele. So in the previous example, the dominant allele was brown. The recessive allele was black. So if we have a dominant paired with a recessive, the hair will be brown because the dominant allele wins. And that is what is expressed in the end. On the other hand, recessive alleles are variations of a gene that can only be expressed if both copies are present, masked by dominant alleles, and the recessive allele is represented by a lowercase letter. So, as you saw in the previous picture, you need two of each, right? You need two alleles per gene. You can have two dominant alleles, you can have two recessive alleles, or you can have one dominant and one recessive. The only time you're going to see the phenotype of the recessive allele is when you have two recessive alleles together. So let's check in. Which allele in each pair is dominant and which one is recessive? The dominant alleles are going to be the capital letters. So capital A, capital B, capital C, and capital D. And the recessive ones will be the lowercase letters. Now let's get into genotype versus phenotype. Genotype is the set of genes passed on from generation to generation, which code for proteins responsible for the expression of a phenotype. So, again, these genes, in the form of DNA, code for RNA, which ultimately codes for the protein, which is the phenotype, as it says here. Then the phenotype is the expressed physical trait for an organism which results from their corresponding genotype. Okay? So, in other words, the genotype is what we don't get to see. We would only get to see if we characterize the genome of the organism. But what we get to see is the result of the genotype, the phenotype. Two individuals can have the same phenotype but have a different genotype. Quite important. For instance, two individuals can have brown hair, but one can have a genotype of capital B, and the other can have a genotype of capital B, little b. Both would still have brown hair. The first individual is homozygous dominant for brown hair, while the second individual is heterozygous for expressing the dominant trait for brown hair. So, heterozygous means that you have both the dominant and the recessive together. Homozygous means that you have both either dominant or both either recessive. In the end, both the homozygous dominant and the heterozygous would show the same phenotype. This is what masking is talking about. So they indeed have a different genotype, but they have the same phenotype, which is brown hair. Homozygous describes a genotype which has two identical alleles at the same locus. capital B or little b. Respectively, they are homozygous dominant and homozygous recessive. The heterozygous describes a genotype which has two different alleles at the same locus, capital P, little p. Due to the masking effect of the dominant allele, a homozygous dominant genotype and a heterozygous genotype will both code for the same phenotype. Again, the definition of masking. Note that the terms homozygous and heterozygous refer only to the genotype. Again, this would be the homozygous dominant. At the same locus, they have both dominant alleles. Heterozygous, at the same locus, they have 1 and 1, 1 and 1. And for the homozygous recessive, at the same locus, they have both recessive alleles. So, use the terms to describe the following. So here are the terms. Which of these would be the homozygous dominant? Homozygous dominant would be big A, big A. Homozygous recessive would then be little u, little u. And then the heterozygous would be big M, little m. As so. Now, true breeding happens when two individuals with the same homozygous genotype breed to produce offspring with a pure genotype. The offspring produced have the same traits as the parents. So, in order to engage in true breeding, the two individuals that are breeding must be homozygous. That does not mean that they have to be homozygous dominant. It means that they have to be homozygous. They could be homozygous recessive. But what we can certainly tell is that all individuals in the next generation that come from that breeding are going to have the same genotype. And if they have the same genotype, they must have the same phenotype as well. So here's an example. Crosses between true breeding plants with purple flowers produced true breeding plants with only purple flowers, which is what you would expect in a situation like that. Chromosomes of a true breeding plant. If it's purple, it's going to look like this. At the same locus, they would have both alleles for purple. And you would get purple flowers. If it's a white plant, then you would have at the same locus, at the same loci, you would have the recessive alleles. In this case, the white color. So if you have both recessive alleles, you express that phenotype. So you express white flowers. But notice that you have to be homozygous recessive. Now, think that if we had a heterozygous plant, would it be purple or would it be white? It would be purple because of the masking effect. Big P wins over little p. Purple wins over white. What happens when we cross the parent plants? So we're crossing a true breeding purple plant with a true breeding white plant. The resulting offspring will all have the phenotype for purple flowers and a heterozygous genotype. How come? Because we have the big P, and the little p, little p. Remember that the purple flower color is dominant to white flower color, so it will overpower the little p, even though definitely the little p went on to the next generation. We will see an example of this. How do we tell what the genotypes and phenotypes will be respectively? We can use a Punnett square to do this. A Punnett square is a diagram used to predict the outcome of any given cross. So these are the rules of how to use a Punnett square. Number 1, you're going to name the alleles and draw a grid that corresponds to the number of gametes that come from each parent. So, as a rule of thumb, you can think that when we're talking about one single gene, two gametes are possible. Okay, so 1 and 2. 1 and 2. Alright? Two gametes per parent are possible. Then label the sides, as shown, with the parents' genotypes. So these are the parents' genotypes right here. It represents the gametes produced by each parent. And lastly, assign the letter at the left and the top to each box. So left and top, left and top, left and top. At the end, you see that all of them are heterozygous in this particular cross. So 100% are heterozygous genotypically, and 100% will be purple by definition of the masking effect. So this is an example of complete dominance. We will talk more about complete dominance and incomplete dominance and codominance and all of that in next week's lecture. But today, let's focus on complete dominance, which is what this example shows. The P generation is said to be the parental generation. So in the case that we were discussing, the parental generation was composed of a purple true breeding plant and a white true breeding plant. So a homozygous dominant plant and a homozygous recessive plant. We already did in the slide before the Punnett square for this cross. So all of them would be heterozygous. All of the ones that come from this cross are going to be referred to as the F1 generation. But what if we take the F1 generation and we put them together again? We will get an F2 generation. So now we're putting both heterozygous plants from this cross into this new cross. And we want to see what the result is. So, which types of gametes can this flower produce? Big P, little p. Alright, let's put them here. Big P, little p. What gametes can this one produce? Big P, little p. So we put it here. Big P, little p. We do the Punnett square. We fill it out. Big P, big P, big P, little p. Big P, little p. Little p, little p. So this is the result. How many of these have a big P? 3 of them. So 3 of them are going to be purple. 1 of them is going to be white, because it only has that little p, little p. Genotypic and phenotypic ratios. Monohybrid crosses from the F1 generation to the F2 generation. As a result of this cross, the genotypes are 2 big P little p and 1 big P and 1 little p little p. Also, we can already see that 3 flowers are purple and 1 is going to be white. The genotypic ratio is the ratio of genotypes in the offspring of a cross, and the phenotypic ratio is the ratio of phenotypes in the offspring of a cross. So here is the Punnett square that we got from this monohybrid cross. The genotypic ratio is then going to be 1 to 2 to 1. What does this mean? We have 1 homozygous dominant. We have 2 heterozygous. We have 1 homozygous recessive. 1 to 2 to 1. Then the phenotypic ratio is 3 to 1 because we have 3 purples and 1 white. A test cross. This can be used when a genotype of a parent is unknown. The second parent is always the homozygous recessive. Now, I want you to think about this for a second. Why would the second parent would always be homozygous recessive? This would be specifically because if it's homozygous recessive, there is only one phenotype and one genotype that can give you that. For example, going with the white flowers. If it's little p, we know it has to be white. It has no other choice than to be white. And the only genotype that will give you white is going to be little p. So if you take a white plant and you cross it with this purple plant that we know can be one of two things. It can be a big P and a big P, or it can be a big P and a little p. So how do we find out? We make a cross. If the plant is a heterozygous plant, it's going to be a big P and a little p, and we would get this result, very similar to the F. We would get this result, all right? And now, if the parent is the other parent, the unknown parent is homozygous dominant, we would get big P, and we would get no white plants. Let's see if you understand this. It ultimately depends on the ratios of the offspring, absolutely. Look at this square here. How many of these plants would be white? We have one white here, another white here. We have a purple here, we have a purple here. So half of the plants would be white and half of the plants would be purple if the unknown parent is a heterozygous. However, if the unknown parent is homozygous dominant, no white plants would be seen. 100% would be purple. So this is how you tell what that parent was before. Here, the variable is this. This is what we're trying to figure out. This is the missing information that we have. If we keep crossing them and no white plants come along, then we can safely say that that plant was a homozygous dominant. A dihybrid cross is a little bit more complicated than a monohybrid cross because we will be looking at two genes. This one is used to cross two traits located at different loci at once. To perform a dihybrid cross, determine the genotypes of the parents and assign them letters. Then FOIL to determine the different gametes produced by each parent. We will see what that means, okay? And next, fill out the Punnett square accordingly. This is what FOIL means. It's a little mnemonic device that says first outside, inside last. This is what it's referring to. In a dihybrid cross, each parent has two traits. So A and B, alright? They can be either homozygous dominant for A, or heterozygous or homozygous recessive for A. Same for B. How are we going to get the gametes that they can produce? We will do it as follows. This A goes first with this B. Then this A goes with this other B. So now we have one gamete that's big A, big B. Another gamete is big A, little b. Then we go with this A. The other gamete is going to be little a, big B, and then little a, little b. So we have four different gametes, as you can see here. Four gametes for each parent that can be displayed on a 4x4 Punnett square. So here you have a complete dihybrid cross. Performing a dihybrid cross between two heterozygous round yellow seeds. Note, yellow is dominant over green and round is dominant over wrinkled. Step 1, determine the genotypes and assign letters. So we said that we are going to be working with round yellow seeds. So both look like this, round and yellow. Because of that, we can tell that this is going to be a heterozygous and a heterozygous pairing together. Now, next step is to get the gametes for each. Determine the gametes produced by each parent by foiling. So big Y, big R, big Y, little y, big R, little y, little r. you can see these are the same gametes. The other parent would produce exactly the same gametes. So now we go ahead and put them on the edges of the square, as so. Then we fill it out. You know how to do this. Big Y, big Y, big Y, big Y, big R, big R, big R, big R. Let's do this one. Little y with big Y, all right? Right here. Big R with little r, right here. Now, the genotypic and phenotypic ratios of a dihybrid cross are going to depend, as everything else does. All right? Now, here we're looking at a dihybrid cross of heterozygous individuals. Both are heterozygous. This is what we get when both parents are heterozygous. There would be 9 round and yellow offspring in the example that we were talking about. 3 wrinkled and yellow offspring, determined by the blue color. 3 round and green offsprings, right here, and only 1 wrinkled and green offspring, right here. The genotypic ratio here is 1 to 1 to 1 to 1 to 2 to 2 to 2 to 2 to 4. This means that we have 1, 2, 3, 4, 5, 6, 7, 8, 9 different genotypes. And it means that we have 4 kinds, 4 repetitions of the 9th genotype, 2 repetitions of the 8th genotype, 2 repetitions of the 7th genotype. But all of these are different, okay? The number, the quantity number just means how many repetitions there are. But each kind of genotype is separated by the 2 dots. The phenotypic ratio, on the other hand, is 9 to 3 to 3 to 1. Why? Because we have 9 that are round and yellow. We have 3 that are wrinkled and yellow. 3 more that are green and round, and 1 more that is green and wrinkled. 9 to 3 to 3 to 1. If you have any questions at the end of this presentation, please feel free to put them on the discussion board. I will happily answer the questions that you put up there. Thank you for listening. Goodbye.